Okay, hello everyone, and welcome to the New York City Neuromodulation Conference Online 2020. It is my pleasure today to host a session on hybrid models for personalization and optimization of tES, a journey across scales. I have with myself a distinguished list of speakers. I will present in a second. Also monitoring the session will be Pavel from Neuroelectrics. He will be managing the technical session. A few notes about how we will work. Chat is open for all ongoing discussion. Please be respectful. You can post questions via the Q&A button on Zoom or via the chat functionality, or you can use Twitter as well because the session is being streamed online in YouTube. We using the hashtag any_webinar. So if you have any logistic concerns, technical issues, please contact the session administrator, Pavel. So the panelists today include Gustavo Deco, who will be talking about an overview of computational modeling approaches to tES, but the focus will be on awakening, promoting transitions between different brain states in a probabilistic state space framework, followed by Amana Berra, physics, multi-scale modeling of transcranial brain stimulation with morphologically realistic microscale cortical neurons. Stephanie Jones will follow, talking about the human neocortical neurosolver, trying to modeling the microcircuitry of cells, a new software tool to study the circuitry of and the origin of EEG and the impact of non-invasive brain stimulation using these microscale models. Fabrice Wendling will follow and will give an overview of neural mass models and hybrid brain modeling, and he will be talking about the mechanisms of action of tCS in epilepsy, the need for hybrid by-inspired brain models, biophysics by-inspired brain models. So I want to give a short introduction to give you a context for the session. I will try to be brief because I don't want to take too much time. So I want to tell you a little bit about the vision of model-driven tES optimization and this idea of going towards model-driven transcranial current stimulation methods, what they call tES 3.0, as I will try to explain. The motivation for what I do, and I think it is interest shared by the panelists, is to try to find solutions for patients that are affected by neurological psychiatric diseases that currently have no good solutions. And the method that we propose is to provide personalized brain therapy by measuring and modifying brain function in a model in a scientifically informed manner. So the technologies that we have in Neuroelectrics include multi-channel brain stimulation systems that are now usable for home use. They can also collect EEG, and they provide an opportunity, but also a challenge, because there are many parameters to be set in this stimulation protocols, many choices for the types of current that you can apply, tCS, tACS, or tRNS, or actually custom waveforms, how many sessions you want to apply, how can you use EEG to inform how to better adjust stimulation protocols, and so on. So this is an opportunity and a challenge, and this is kind of the rationale for my interest and for the interest of our company in developing solutions. So the challenges include how to model electric field under tES. How can we model this accurately? But once it is known, this is known, how can we model the effects of this at the cellular and system level? And beyond that, once we know how that happens, how can we use it to actually achieve therapeutic outcomes? And the idea behind hybrid brain modeling is to bring this physics and computational physiology together and to provide this sort of an ignition of new technologies and new solutions, as I will explain. So from the point of view of biophysics, we are now at a certain level of maturity in measuring, in quantifying the electric fields that we can generate with brain stimulation. Both the tES, TMS, and ECT are provided here as examples. So we can do this, and we can do it at a personalized level, informed by MRI neuroimaging. And this is something that it's quite mature. We still have challenges, but I will not go too much into this right now today. At the level of mechanisms, we have a simple mechanistic approach that at least we use today when we provide solutions to researchers and to clinicians, which is to use this lambda E model that assumes that the effect of the electric field can be modeled to first order, at least, as the dot product of the electric field with the direction, the orthotropic direction of cortical neurons, pyramidal neurons that are aligned nicely or in a nice and orderly fashion. This is clearly a simplification. This is a starting point, and we need to go beyond that. We will be hearing about some of this work by Aman in this regard, which I'm excited to hear about, how to better model these effects, the impact on the transmemory potential, and the nice biophysics of all this problem. But even if that is solved, then we have the next question, which is to try to understand how the effects of these electric fields interact with populations of neurons. And in particular, one challenge is to explain the great sensitivity of neural populations to very weak electric fields, which affect only at the very sub-millivolt level the transmemory potential. So this is one challenge for the field to explain how this happens. And I think it's a very interesting one. So once we have basic models, we can start talking about optimization. And today, the way this is done is by providing a simple, simplistic view, which is to say, I want to stimulate this area, I want to excite this region, I want to inhibit this other region. This can be done at the by using some maps on MRIs and so on. Once that is specified, then we can build a personalized brain model for a subject. And based on that, also taking into account if there are skull defects, implants, and so on. This is something that we can already manage. We can provide a solution, which is in the sense that we're providing the best montage in terms of electrode positions and currents that, respecting some constraints, gives the best match of the desired electric field with the produced electric field by the montage. So we're talking about things here at the level of electric field, which is not physiology yet, but this is what we do today. We can go a bit beyond that by using neuroimaging data, for instance, from resting state functional connectivity MRI, and we can look at the networks that are associated to certain functions in the brain and produce montages that match the desired interaction with these brain networks. Okay, so this is a higher level of sophistication, I would say, than the one that I was describing before. And this is something that can be done today. But again, the language that we use is we're going to apply an electric field in a given part of the cortex because we want to excite or inhibit this area of the cortex. This is the level of sophistication. So in tES 3.0, the idea would be to bring this physics with a more mature and quantitative physiological view of what's going on, right? So this work began, I think, at least as far as I am concerned, it began in 2013 with this nice work by Isabelle Merlet and Fabrice Wendling and others, in which for the first time, the biophysics of the electric fields on the brain was brought together with neural mass modeling and the first version or one of the first versions of a hybrid brain model. And in this, I would call pioneering paper, it was already shown that one can use this approach to try to understand how tCS in this case can interact with the whole brain and how you can see resonance effects and so on and so forth. So this is the beginning for me of hybrid brain modeling. We have gone some ways forward from since then. One of the questions that remains, though, is how to match the biophysics and the physiology across scales. So from neural models that can be physiological models, Huxley, with neural population models such as Janssen-Reed. So this bridge between these two scales is something that we need to understand better. We need to work a little bit more on that. We have been working in my team on, for instance, developing a laminar version of neural mass models that can at least make a better match, a closer match with physiological measurements, for instance, from probes, brain probes, try to understand the dynamics of electric fields, the currents and the CSDs. And this is something that we can use to try to develop better neural mass models. Yeah, and the nice thing is that you can match what these models produce with actual data, which is of course very important. So putting all these things together, we arrive at this idea of a hybrid brain model in which we have some level of representation at the mesoscale of neural mass models. By mesoscale, which is a word I will use repeatedly, I mean the scale between single neurons and the entire brain. This is the level at which we are working with hybrid brain models, but we need to understand how to go from micro to meso and then to macro. And one of the ways to do that is to use neural mass models coupled across each other, placed at different points in the cortex, coupled by some connectivity matrix that can come from structural connectivity, for instance, from diffusion tensor imaging. But once this is done, we can produce something that can be contrasted with data such as EEG in a realistic fashion, right? So the vision is to create personalized models, hybrid brain models that include the biophysics, the MRI, the anatomy of these patients, but also that are able to ingest EEG data and other data from them, and to produce a personalized version of their brain that they can be used to simulate the action of tES and to use that for optimal treatment, and which can also be used for adaptation as time goes by collecting further data from the patients over time. So this is the vision of tES 3.0. I want to mention that we are about to start a project with Fabrice and Fabrice, a synergy ERC project exploring this hybrid brain modeling approach in the context of epilepsy, trying to traverse the scales in modeling, and actually by year 4 to produce a solution for a clinical trial for epilepsy patients. And we're actually hiring. So if you're interested in this area, please do visit this website that I provide at the bottom. So to summarize, from tES 1.0 to tES 3.0, I just try to give a high-level view of what's going on. So from the point of view of what type of electrodes we're using, the target definition approach, the brain models that are used, the optimization function that one is using to find the best solution, and the methods. From tES 1.0, which began with the work of Michael Nisha and Paulus, using sponges, defining targets, using basically the 10-10-20 system without much of a physical brain model behind it, and optimization. We went to tES 2.0 using small electrodes, then the target definition using the parcellation or a user-defined cortical model, talking about networks already, we're doing that already. Brain models which include the physics of the conductivity of currents in passive tissues. The optimization function used right now, as I mentioned, is defined by the electric field desired, which is kind of crude, but that's what we do. And the optimization method is algorithmic now. In tES 3.0, the major steps, I would say, are going to be defining optimization functions in terms not of electric fields, but in terms of the functional change that one desires. And this will have to deal with the issue of plasticity, which is something that we'll probably hear about in the talks in a moment, and using these hybrid brain models. So some of the questions that I think today I would like to hear about, and I'm sure I will, is what are the best modeling approaches? What are the appropriate microscale models that we should be using in the context of tES optimization? How can we translate them into mesoscale and then macroscale models? How can we meaningfully create networks that integrate these models into hybrid brain models? About the mechanisms of interaction, what are the effects at mesoscale? How can we understand the effects at microscale, which we're sort of have a first grasp on, but how can we go to mesoscale and then to microscale? And then, of course, how to use this knowledge to force transitions between brain states by external stimulation, and how these transitions between brain states can result into therapeutic plastic changes, which is what we do in tES. So I want to thank a long list of people here, including people from my team and my collaborators, which are really a fun part of my scientific life. And without much more, I just want to introduce the first panelist, which is going to be Gustavo Deco from ICREA UPF in Barcelona. And he'll be talking about awakening, promoting transitions between different brain states in a probabilistic state framework. So Gustavo, I hope you can, I'm gonna release the screen and back to you.
Okay, thank you, Julio, for the introduction and for the overview. I start sharing my screen. Can you see my screen? Yeah, okay.
Yes.
So the title was already mentioned. It's about using whole brain modeling for awakening, and with awakening I mean in a rhetoric, poetic way, how to force a transition between different brain states. So the motivation is cartoonize it in this slide. The first slide, I mean, as Julio mentioned in his introductory talk, I mean, the goal is really to see and to discover the necessity of using mechanistic whole brain modeling in order to predict the effect of stimulation. That could be electrical stimulation, the TMS, or tES. And even more, it's not only just to predict the effect of the stimulation and to model that, but perhaps to use then the mechanistic models, mechanistic whole brain models, in order to design or to discover and predict where to stimulate in order to target a particular state. This is also cartoonized in this second, more philosophical type of slide. I mean, the idea is following. We have, let's say, a brain in a given state. I put it here just healthy, just to put something, let's say this is our source state. And this is characterized by a certain dynamic that we can describe with a dynamical landscape. Then if we have another brain state, or let's say, for example, the resting state characterizing a particular disease, then we can assume and we can quantify and measure that the dynamical, the underlying dynamics is distorted, and this is characterized here with this shifting of the landscape. And of course, one temptation is to design or to use modeling in order to design a stimulation such that we recover the original working point. And that is actually extremely complex, because basically in a distorted landscape, we have to design a control of the trajectory for a particular trajectory in order to recover this particular point. Our philosophy is totally different, and it's schematized in the last column. The idea here is that we have this distorted landscape, this distorted dynamic, and we will try to study with which type of stimulation we can shift the dynamic to a state which is our target state. In this case, if it is a disease, we want to go in the direction of a healthy state. And then we will, because the stimulation is just shifting this dynamical landscape, then we hope that by homeostatic recovery, by plasticity, the system will self-organize to try to adapt to this new situation. So the goal of the stimulation in our study is really not to control a given trajectory or to control a given particular point in our dynamical landscape, but to control the landscape, to distort the landscape. That is the motivation. So I divided the talk in 3 parts. First, I will say a couple of words about the whole brain model that we use. Then, of course, the second element that we need is a robust definition, a quantitative definition of the brain state, because we want to promote the transition between source brain state and a target brain state. And of course, in the third part, we will see how we can study the forcing of such transition. So a couple of words about whole brain models. As you know, it's a field which is relatively well established nowadays. And the key idea is basically to couple the dynamics associated with the local level, which could be a population of neurons or a given brain area at different level of parcellation. And the coupling is defined, of course, through the anatomical connections. In most of the cases, that can be gained, for example, in the case of human, in a non-invasive way by DTI tractography, and that is translated in what we call the structural connectome, defining the connectivity, the anatomical connectivity, linking those different local dynamics. And the main idea is to study and to fit with different optimization and theoretical methodologies, the functional level. So in this case, for example, the most simple one is just take, for example, also MRI, in this case fMRI, and calculate the correlation at the functional level in order to identify, for example, under resting state condition, the different resting state networks. And this is practically compile it in this functional connectivity matrix, the functional connectome. And the idea is that we are able to reproduce this functional connectome. There are many models coming from many different labs, and they have many different assumptions. The main ingredients that where we have the freedom to modify the different aspect of the model is by the local dynamics. For example, you can assume that the local dynamic is really extremely simple, for example, just noise, or you can assume that the local dynamic is extremely well detailed at the level of spiking neurons or reduction using mean field or neural mass model of the spiking level, which is mainly characterized by the non-oscillatory behavior. So it's a kind of asynchronous regime, so very similar to the first scenario, or the third scenario where you introduce on purpose, because it's also biophysical realistic with different type of neural mass models like Kuramoto, Wilson-Cowan, Fishman-Agumo, etc., oscillations. The idea or the model that I will use here is somehow a kind of syncretization of those models, in the sense that we unify the asynchronous and the synchronous scenario, so the oscillatory and the noisy scenario. And this is schematized here. And the key idea is that we use for the local level a model, a kind of neural mass, but even much more reduced, because we use the local form of a Hopf bifurcation. It's a horrible name, but the idea is very simple. It's the most simple way of describing mathematically the transition from a fixed point, which in our case, because it's also distorted with noise, means that from a noisy scenario to a full oscillatory scenario. This is what it's a Hopf bifurcation called. And this is regulated by a local bifurcation parameter that we call A. And of course, if this parameter is very negative, like in this case, then you have just noise. If it is at the edge of the bifurcation, you have a mixture between noise and oscillation. And if you are in the positive range, you are very oscillatory, like in the case of the Kuramoto model. All the other ingredients are common to the other models. So we interconnect and couple those local dynamics through the neuroanatomical matrix in form of PTI tractography, and try to explain different aspects of the functional dynamics. Of course, and it's very important, and we have not the time to go into the details, but at least I just want to mention that. I mean, we fit the model by using not only the traditional functional connectivity, which is a kind of grand average static spatial measure that we already mentioned, but we take different spatial-temporal aspects. In this case, for example, we take the seminal idea introduced from the lab of Vince Kalun by Allen et al. Many years ago of the functional connectivity dynamics. The idea is that you study now the functional connectivity, but in a relatively small window, and then you slide the window, and basically you compile in the functional connectivity dynamics the statistic of this change, not only in space, but also in time. And this is an information which is extremely relevant, and we can see that here very explicitly. In this case, you see these two measures. Let's concentrate on the right panel, the functional connectivity and the functional connectivity dynamics. This is just the error with empirical in form of the Kolmogorov-Smirnov distance. So the bluish, the better is the fitting. Here is the contrary. It's just the Pearson correlation between the functional connectivity. So the brownish, the better. And we have these two parameters, the global coupling, regulating the global conductivity, and the local bifurcation parameters. In this case, as a matter of simplification, we assume that it's the same for all nodes. And as you see, there are a very particular region, which is around here, where we have a perfect fitting of the functional connectivity and of the functional connectivity dynamics. So in this point, practically at the edge of the bifurcation, the functional connectivity is very well fitted, so the correlation is very high, and the difference of the functional connectivity dynamics, or the error in form of the Kolmogorov-Smirnov distance, is very small, so that is a very narrow and very good fitting that you cannot obtain in other regions. For example, in the negative regions, so in the pure asynchronous scenario, or in the pure oscillatory scenario. So meaning that in this unified model, you get a very constraining fitting of spatial and spatio-temporal aspect exactly at the edge of the bifurcation. So exactly at this mixture between noise and oscillations. So this is the whole brain model that we use. Has also a good, I mean, it has a very good property, as we have seen. I mean, can fit very well empirical data, fMRI data or EEG data. And also it's very easy to perturb, to stimulate. We will see how we do that. But before going to that, I mean, let's see how we can describe a brain state from a quantitative point of view. And this is compiled in this slide. The idea is very simple. Was explained in our paper at the tES last year. It's a method introduced originally by Joanna Cabral many years ago. The idea is very simple. It's a kind of a spatio-temporal functional connectivity, but run at the phase space. So meaning we calculate the phases using the Hilbert transform of the fMRI signals. For each time point, now we define a matrix, which is basically a coherence matrix, defining the cosinus difference between the different phases. So it's like an instantaneous, practically state of the synchronization of the system. And then we do this and we slide this point. So we describe this across all the points. The second step is, well, there is an step in order to reduce dimensionality, but you can do this optionally or not, depending on how fine is your parcellation. You, instead of taking the matrix, you take just the leading eigenvector, which capture over 85% of the variance. And then basically you clusterize these matrices or the leading vector of those matrices. And by clusterizing, basically, you find that the system is moving between different centers, different states, and you are describing those states. And even more than you can, and this is the reason why we call it a probabilistic state space, because you can calculate of these substates that we call metastable substates. You can calculate the probabilities, or you can calculate the transition probabilities. And basically this probability expression is defining a given brain state. So if this is resting state, this is the probability description of resting state. If this is the resting state of a disease case, schizophrenia, then this will be the corresponding probabilistic definition. So let's now discuss very briefly how we can study the forcing of a transition between those brain states. So now we have all the elements. We have the model, which are fitting the data, and we have a very good definition, quantitative definition of a given brain state. And just to be pedagogical, and as we have shown in that PNAS paper, in this case, we work with data from the lab of Slav, basically healthy participants, which are under resting state condition, or wakefulness, or under sleep condition. In this case, I just took the deep sleep, meaning the N3 stage, and we fit the model. So first, we characterize it with the probabilistic state space, the probability profile of metastable substates, and with the model, we fit that particular probability profile. And as you see here, this is not a cartoon, these are really the real data. It's a set of 18 people, if I remember properly. And what you see is that you have to detect here two things. At the empirical level, resting state or wakefulness is very different from sleep, from deep sleep. Statistically significantly. In particular, one of the superstates, which is statistically very different in one case or the other case. So we can distinguish with this probabilistic methodology the two brain states. And the second remark that's important to see here is we can fit with different models, of course. So we can design a model, which is a Hopf model in this case, of wakefulness, and a model of sleep. And then you have to see the similarity between these two profiles in one case and between these two profiles in the other case. So now that we have the models and the goal, so the idea is to take a model in silico, for example, of one of the source state. Let's assume that we go to the sleep state, and then we want to awake, so meaning we want to find a stimulation such that we perform a transition to a given target state. And let's say that the target state is wakefulness. So we want to provoke this awakening in silico and to study in which region and at which level we need to stimulate in order to provoke that transition. As you see, these two main ingredients, the mechanistic model in order to exhaustively try in silico all the possibilities, and second, the robust definition, a quantitative definition of the target, of the source and the target state. As I said, in this case, wakefulness and sleep, or you can go in the next slide, you will see the other way around, from sleep to, from wakefulness to sleep. The idea how we perturb, in this case, the model, is artificial because we were only interested not in a realistic biophysical stimulation, but in the effect of the stimulation per se. And it's just by moving locally in a bilateral way, the local bifurcation parameter. So meaning by promoting noise or by promoting synchronization, oscillations in one particular region. And that we can do with different level of intensity and then do region by region exhaustively. Of course, this takes a lot of time, but given that we are able to do this in silico, we send this to the cluster for a couple of weeks and we get the result. The colors means how well we are achieving the transition, meaning how well we are fitting, so bluish is a good fit, how well we are fitting the target state, meaning that if we are reaching here deep blue, means that for that particular region, for that particular level of stimulation, we are forcing a transition. So let's see with a little bit more detail. These are the different way of stimulation. As I said, you can promote synchronization or promote noise, and you can go in one direction, sleep to awake or awake to sleep. And you see then that the level here, the color level, everything looks very blue, but you can distinguish more in a fine way. I mean, which are the regions which are more prone to be stimulated in order to promote that particular transition. So meaning that we can use this framework at least to detect the sensitivity of the different nodes for promoting a given a specific transition. Just to finish, I want to mention very briefly, it's not the main subject of my talk, but because it's a conference on neuromodulators, I wanted to say a couple of words about that. We can apply exactly the same philosophy of stimulation, but now not thinking on electrical, magnetic, or type of stimulation, but on pharmacological stimulation. And this is really very interesting because we need to expand the models in order to include the pharmacological ingredient that we are adding to the whole business. In this case, we study the stimulation of serotonin receptors. Basically, we are using data from Robin Carhart-Harris in the Imperial College that uses, as you know, LSD, psilocybin, and different psychedelics in order to see the resting state, how the resting state changed in the case of healthy individual participants when you excite serotonin receptors. And so in order to model that, we include the density of serotonin receptors that we gain through PET. So meaning that now we are fusing DTI, fMRI, and PET information in the model mechanistically. And basically the density of PET receptors will regulate the level of the gain of the local, we use in this case a neurodynamical model excitatory inhibitory population at the local level, and we manipulate the gain. So just the main idea is that if you introduce this new parameter, then you can describe the change introduced in this case, for example, by LSD. This is the level of fitting, so the smaller the better. And by using this extra information, you can account for the pharmacological effect. Of course, if you compare with the shuffling version of the PET, you are not so good. Or if you compare with the excitation of different type of receptor or different type of manipulation, because the LSD and even more psilocybin are very specific for one type of receptor, which is the 5-HT2A. This is the one that we consider here. And if you consider the PET information of the other receptor, you are not so good. So meaning that the model is working pretty well. Okay, I conclude here. I think the take-home message is the necessity or the utility, or the promising utility of whole brain modeling, of mechanistic models, in order to help us to design and to predict the effect of particular stimulation. This is, I think, the first, and the stimulation could be pharmacological, electrical, or magnetic. And the second take-home message is that the main idea or strategy is to study how to promote brain states, or how to change landscapes, and not how to control particular points or particular trajectories. I think I finished here. I think I wasn't on time.
Thank you very much. Thank you very much, Gus, for a very exciting talk. I want to open the floor now for questions from the audience. If you have any questions, please use the Q&A or the chat. Let me see if I have something. Let me see. I don't have any questions, I think, yet. So one question I wanted to ask you is regarding this probabilistic state framework approach. I mean, there is clearly an interaction between the modeling approach used and the state framework that one can derive from it. And this interaction is very fruitful. But do you think that this classification, because we need a grasp on how to talk about brain states, do you think this is the right way to go into this probabilistic state framework description of what the brain is doing?
No. I think it's a good proxy, to be honest with you. I mean, I searched in the literature really about a good definition of brain states. And I was really disappointed. There are not many. I mean, we have a mainly qualitative description of brain states. We know that there are different brain states. We know clearly that sleep is very different from wakefulness. We know that psychedelic state is very different from non-psychedelic state. We know that schizophrenia is very different from healthy state, but we don't know how to catch that. In fact, in the resting state community, they try to catch that at the beginning in the most simple way, by functional connectivity. But the functional connectivity is not so powerful. Then the second candidate is functional connectivity dynamics. It's good and it's improving the characterization, but it's also not so powerful. So therefore, we try to go a step forward and be a little bit more detailed. And I think the PMS, or the probabilistic framework, is in that sense a better, a more robust quantitative description, or more sensitive description of different brain states. Of course, I am convinced it's not will not be the last proxy. I mean, I only think it is one of the best approaches that we have until now, but we are trying to improve that and be even more sensitive and more tailored.
Okay, thank you very much. Are there any other questions from the panelists or from the audience?
Yes,
I have more questions. Yeah, we have a question right here, actually.
Yes. Thank you, Gustavo, for this very nice talk. I really enjoyed it. And during the introduction of the talk, I thought about some neurological disorders like absence epilepsies, or obsessive compulsive disorders like Tourette syndrome. And a very particular feature in these disorders is that when the attention level is changing, it might completely change, in fact, for instance, the frequency of seizures. I'm talking about absence seizures. And this is very often reported by neuropediatrics when, especially in children, when the attention is focused on a task, the patients experience less seizures, okay? And it's also the case in the Tourette's syndrome, when the patients are very well concentrated on a task, they have less behavioral, in fact, symptoms. And I was wondering whether this might be due, in fact, to a change of the dynamical landscape. You know, what you said is that instead of stimulating or modifying the system itself, we may modify, in fact, its landscape in order to change, in fact, the state of the whole system. So does it, I mean, according to you, is there a link?
But that's resonate with my way of thinking. Yeah, absolutely. I mean, in that case, I would interpret like the attentional condition, the attentional task, as the stimulation, promoting a change or a good change to a landscape, to a dynamical landscape, which is fruitful. And the more you stay there, I expect, I am convinced that then homeostatic plasticity will help the system to be there without that extra help at some point. So that is it's exactly the same. So it's another type of behavioral stimulation. Let's say.
Okay. Thank you.
Thank you. Thank you. I think Stephanie, did you have a question as well? I see you raise your hand.
Yes, thank you, Gustavo, for that very nice talk. I really enjoyed it. I had one question about, one of the things that I'm going to talk about in my talk is the importance of layer specificity in the information flow between different cortical areas. And I missed in your talk if you include that level of specificity and what kind of the current state of the art is and what we can infer from the tractography information that you're including in your models.
Yeah, I mean, that's a very good question. I mean, and we are trying to use now laminar, very, very simplified laminar models because we want to introduce the effect, which is not relevant in fMRI, but is relevant in imaging, of very particular bands, frequency bands, in the feed-forward and feedback transmission of information across the different brain areas. And as you know, there are many evidences. There was fragment during the last decade, I would say, about the specificity. I mean, how beta and alpha are very specific, communicating one and the other direction. And this can be captured very nicely with the laminar model. So meaning that we didn't start with the laminar model just as a matter of simplicity, because we have so many other problems. But I am absolutely in favor and convinced about the necessity of laminar model. And the next step will be this very reduced type of laminar models, basically with two stages, but able to switch from alpha to beta and gamma transition. So yeah, I think the final model will be a laminar model.
And can we get that information from the tractography?
No.
Do we have that level of detail yet in the technology?
No. Actually, in monkeys, we have that information because we have the specificity of the interlaminar connections. For example, the lab of Henry Kennedy in Lyon. Those are very massive tract tracing studies. With non-invasive technique like DTI, at the moment it's not possible. Perhaps using high field at some, I mean, because we have access to the laminar information, at least in the functional MRI, it could be that at some point we could get a specific DTI information about that.
Okay, thank you, everyone. I think we have more questions, but in the interest of time, we should continue.
The screen. Yeah.
There you go. Yeah, thank you. So The next talk will be by Aman Aberra. He will be talking about E-fields in morphologically realistic microscale cortical neuron models, trying to give us a better grasp of how the electric field is altering the electrical properties of neurons.
Can you see my screen? All right, I see some nods. Yeah, so thanks for having me. My name's Aman, as Julia mentioned. I'm here in Durham, North Carolina. I'm a PhD candidate at Duke University, working in the labs of Dr. Warren Grill and Engel Peterchev. Yeah, and I'm excited to talk to you about the work I've been doing for the last few years, trying to basically combine the information we can get from these detailed, anatomically accurate ball and conductor models of human heads with biophysically realistic cortical neuron models. Yeah, so I think there's been a lot of really great background already, so I won't spend too much time on that. But the basic kind of motivation for what we're doing is trying to understand in these transcranial brain stimulation modalities, and I'll be talking mostly about transcranial magnetic stimulation here in the top, and transcranial electrical stimulation, or specifically tDCS, transcranial direct current stimulation. Whatever modality you choose, these stimulation modalities generate electric field distributions in the brain, which we have now fairly detailed models. And other sessions in this conference are talking about these electric field models and the techniques we have to simulate them using MRI imaging. But the key question is, how do we go from that electric field distribution and understand the neural response? And going a little further, not just in terms of the ultimate goal of getting the physiological response, but understanding what happens at the single cell level. How does the electric field couple to these neuron models, or to these neurons, which we're in a model? And how do the spatial and temporal parameters of stimulation determine that response? So that's what I'll be talking about a bit. So just as an outline, I'll be talking about the multi-scale model we've developed that, like I said, combines these biophysically realistic single neuron models with MRI-derived head models to try to understand how and really capture in a single model the spatial and temporal features of the stimulation and the response. And then I'll try to briefly go through two short stories on using this model for TMS, which we published last year, and then some currently unpublished but preliminary work applying this same model to tDCS. And then I'll conclude. So first, I just want to introduce the neuron models, and what we're using are essentially morphologically reconstructed models drawn from imaging and brain slices that allow us to then compartmentalize the morphology of the neuron and represent the electrical activity in the neurons using cable theory, which essentially breaks up the neuron into little circuits that we connect and then embed with nonlinear ion channels to simulate the electrical dynamics. So the advantage of these models for this type of this study is that they allow us to both capture the spatial extent of the neurons, which determines basically where they're positioned in the electric field and how the electric field couples to the model, as well as the temporal dynamics at a microsecond scale. And this is down to the level of detail of the ion channel models, which we solve numerically, and typically are these Hodgkin-Huxley type models. And importantly, we can couple these models to electric field distribution, as I mentioned, where I think typically, or in more simple models, we might represent the cell as a single node. We lose this detail of the full axon, the full spatial extent of the neuron, which, as most people know, is very important to determining basically the integration both of intrinsic activity, but the integration or the effects of exogenous electric fields on the neural response. So what we worked on was basically adapting a library of cortical neuron models published by the Blue Brain Project in 2015, which essentially drew data from brain slices in juvenile rats. And we adapted them by essentially scaling them geometrically and incorporating their axon to adapt to the properties of adult human cortical neuron models. So this is showing the models that we extracted for the studies that I'll be showing today, which are include interneurons in layer 1 and layer 4, which are the neuroglialiform cells and the basket cells, and pyramidal cells in layer 2, 3, layer 5, and layer 6. So this is showing not only that we have each layer represented, but we have multiple virtual clones represented in each layer that are essentially stochastically varied morphologies of each cell type or each class of cell. And one advantage of this is that the data that's used to generate these models is drawn from these low-level neurobiology studies, as opposed to being fitted by TMS or tES data. And just to highlight, one of the major changes we did was incorporating myelination, which develops with age and has higher levels in humans and adults, and it also has regional variations in the brain. So in this first study, we characterized and validated the models with a range of stimulation modalities. I'm just going to focus in the interest of time on the response to subthreshold uniform fields. Julio showed this figure, I think, in the introduction. But and the first thing this kind of highlights here is what we're doing here is essentially applying an electric field downward in the direction of the cell. And just for reference, the somatodendritic axis here is going up and down on the page, which is parallel to the cortical columns that the cell would sit in. And what you see already, and this has kind of already been established previously, is this biphasic distribution of the polarization. So in the color, we're showing the polarization across the cell. Where the field points down, you get depolarization. Where the field leaves, essentially the current essentially is leaving the cell, you get depolarization, and where it enters, you get hyperpolarization. And the soma, which is generally what we focus on, sits somewhere in between, and depending on where it is in between the two poles of the cell, you get some depolarization or hyperpolarization. So the first thing we did was compare this to in vitro experiments. One key one was a study done by Radman et al. In 2009 that showed at these low intensities applied that are relevant to tCS, you're in still this linear regime of polarization. And they quantified the polarization length, they called it. It's kind of a weird metric, but essentially it's just the polarization in millivolts per volt per meter of the field. And they quantified this across cell types and found that you get highest polarization in the largest cells, these layer 5 and 6 pyramidal cells, and this is in the soma. And kind of much lower polarization in interneurons, but a range basically where the same direction can produce both depolarization and hyperpolarization. But what we also were able to do in the models that they weren't able to do experimentally in that study was quantify the polarization in both the axon and the dendrites with these more detailed models that capture the actual axonal and dendritic arbors. And what we find is you get significantly higher polarizations in the axon and dendrites, particularly at the terminals, where the coupling of the field is much higher than in the cell body. So you get closer to a millivolt, for example, in the layer 2, 3, and layer 5 pyramidal cells, which matches more recent study that actually measured axon terminal polarization and axon cut ends in mouse slices of layer 5 pyramidal cells. So this is one key kind of finding that we can already gather from these more detailed models is that despite the soma being the kind of key area we look at because of its importance for integrating input signals, these subthreshold transcurrent electrical stimulation techniques are actually producing much stronger polarizations in the dendritic and axonal arbor than in the cell body. So that's just kind of introducing these models and a brief look at how they behave at a single cell level. The next step is to couple them to the electric field distribution generated by these transcranial stimulation techniques. So the way we do this is pretty well established. We build head models using MRI imaging, and I'm particularly using the SIMNIBS package. And then the next step was to basically capture the positioning of the neurons relative to their layer within the region of interest. In this case, we focused on the motor cortex. So here I just am showing the interpolated cortical layer surfaces, which I use to then populate that brain model with the neurons automatically. And the outcome of it, just to kind of cut to the chase, is this population model here focusing on M1 and the hand knob region here. This is the kind of canonical omega hand knob representation or hand representation. And you can see the same neuron models now represented in all the layers. And the key thing with positioning these models is that we've aligned the somatodendritic axis with the cortical normal surface, normal vector, which matches the histology. And you can see here basically just some pullouts of the layer 2, 3, and layer 5 pyramidal cells with the axons in red, the apical dendrites in blue, and the basal dendrites in green. So next, we need a couple of these electric fields. So here we've simulated, for example, a posterior anterior directed field, typical electric field distribution, you can see. But what we can do is basically at the cell level, couple the electric field distribution, this non-uniform electric field distribution now, to the compartments of the neuron and neuron stimulation software. And here for TMS specifically, because it's a magnetically induced field, we have to think a little bit more carefully about how to do that. So in the interest of time, I'll move past that, but essentially we use this approach called the quasi-potential approach that's you can find in this reference by my colleague Bo-Shuo Wang. And finally, we can get what we started out to do, right, which is to get the neural response. So one of the things we do for TMS, which is a super threshold stimulation modality, is to look at the activation threshold distribution. So now what this model allows us to do is go from this electric field distribution to a layer-specific quantification of the response of the neurons, in this case, in this motor cortex region. And we can extend that and do the same thing for tCS. So on the left here, I'm just showing simulation of the polarization distribution now. So for each location in this layer surface, we're looking at a color that represents the steady state polarization of the cell body of, in this case, layer 2, 3 pyramidal cells on the left. And on the right, the threshold of those same cells for activation by TMS with a monophasic pulse. So this is kind of pulling together the whole platform, allowing us to capture both the spatial distribution here in the top row of the electric field and the temporal waveform that's being applied. And then finally, we can extract whatever metric we need. For subthreshold modalities, polarization is right now kind of the most salient, whereas with superthreshold, we're looking at what's the threshold of activation, where is that activation occurring, and what elements are activated. So using this model, the first thing we did was study TMS, which is my area of focus, and specifically ask the question of where does TMS activate neurons for M1? You've already seen a little bit of that. And then which neural elements are actually activated. So one of the kind of motivating questions here is this question in the field that's still somewhat controversial, which is where actually does activation occur when we apply TMS, for example, in M1, where most studies are focused. So there's one kind of camp that has argued that the gyral crown and lip, where the electric field is maximal, since the electric field falls off with depth, is actually the site of initiation of action potentials. And this is based on, for example, studies that combine FEM modeling of the electric field with basically correlations of the electric field magnitude with changes in motor threshold as the coil orientation or position is changed. So as that coil position is changed, you get a change in electric field distribution and a corresponding change in the motor threshold. And you can correlate that to find where does the electric field change best explain the changes in the physiological response. And these approaches, in this case, basically give you predictions that say that the field is most explanatory here in the superficial regions in the gyral crown. On the other hand, PET imaging has suggested much more deep activation in the sulcal walls, where we find that the normal component of the electric field is maximal, and has led to the development of this idea called the cortical column cosine theory that basically uses a simplified representation of neurons and says the neurons where they are parallel to the field here in the sulcus couple most strongly, whereas in the gyrus they're perpendicular, they don't couple at all according to the cosine, and we get less activation. So you can already see with these models, and we're kind of just going to illustrate more clearly here, the threshold distributions show that activation thresholds are much lowest at the gyral crown and lip in the model and fall off very rapidly with depth. where the field drops off rapidly. And this not only agrees with that first experiment I mentioned, but also two more experiments that have come out more recently using similar models or similar methods also seem to confirm that we're getting this gyral superficial activation matching the model. So what does the model say about why that might be? Just briefly, when we look at correlating the electric field magnitude versus correlating the electric field normal component with thresholds, we find much stronger correlations with the magnitude, shown here in this top row. And the reason is because activation, as I said earlier, is occurring at these axon terminals, which, according to cable theory, are proportional. Their polarization and activation is proportional to the magnitude of electric field. So what's shown here is the activated axon for each neuron in this slice of the layer 2-3 pyramidal cells, and this layer 4 interneurons and layer 5 pyramidal cells for, in this case, a PA directed field. And what happens when we then reverse the field is you reverse and flip essentially the action potential, the site of action potential initiation within these axonal arbors. And the reason why that's interesting is here I'm just showing a small piece of data from experiment in 2006, but this is a pretty commonly found finding, which is when you apply TMS in a posterior anterior direction versus an anterior posterior direction, this just flipping of the field produces about 2 to 3 millisecond difference in the latency of motor evoked potentials. So even though the field distribution is identical, we're seeing a change in the physiological response, which might suggest a change in the location of activation. And what we see in the model here, I'm showing a slice of the hand knob region and the threshold differences between AP and PA. So where you see red, there's more activation in the case of anterior posterior stimulation. And where you see blue, there's less activation of anterior posterior and more activation of PA. So here in PA, where we get a short MEP latency, we're getting more activation here again in this kind of posterior lip region. Whereas AP, which produces a longer MEP latency, is producing this anterior shift of activation. The reason this might be interesting is this matches up pretty nicely with what's seen in the cytoarchitectonics of monkey M1, which likely is homologous to human, which basically this study found that you get lower thresholds for activating hand motor neurons directly in the sulcal wall, and that the direct projections to those hand motor neurons are more likely found in the sulcal wall than in the gyral crown, which is this kind of middle part here. So just to conclude that that section, what we're finding with TMS is that in this multi-scale model is that TMS of M1 predominantly activates neurons in the gyral crown and lip. This likely even includes, and it points to a more dorsal premotor cortex actual potential activation, which has been argued in a recent preprint that used neuroimaging data to study the actual response to TMS as well, and is finding this more dorsal activation that we're seeing in the model. What the model is able to do is explain this is because of the high correlation of the thresholds with the magnitude of electric field due to activation initiating at the axon terminals. We also can find layer-specific effects or the layer-specific response. So I didn't get to go into it as much, but layer 5 pyramidal cells happen to have the lowest thresholds, followed by layer 2-3 pyramidal cells and the large basket cells. And finally, what we're able to show in this model is that reversing the current direction can cause an anterior shift in activation, which might explain these effects, the physiological effects, which can't be explained with simple, just the electric field models alone. So last, try to go through pretty quickly, applying this model to studying some questions about tCS. So one thing I want to introduce here is one way, or one thing that might be useful to understand is how does the cell polarize differently as we change the direction of the field, because this is really relevant to how does the same, for example, same cell type, how might it polarize differently if it's found in the cortical gyral crown versus in the sulcus. So here I just want to kind of orient everyone to the next plot. So what I'll be showing is polarization across all the directions in the sphere. The directions are all indicated by these little markers. So for example, the fields going out of the page can be represented on the front of the sphere here versus fields going into the page on the back. So just to move to the plot, hopefully that was helpful. This plot now shows the steady state polarization of the cell body of this layer 2, 3-pyramidal cell. And the map essentially is just a map projection, a 2D projection of that sphere I was showing in the previous slide. And what you can see here is before I'd showed the results comparing to the experimental in vitro study for polarization of a downward electric field, which is represented here in the bottom of the map projection. But what we see is, first of all, obviously, if you flip the field, you get a flipping in the polarization. You go from depolarization in red to hyperpolarization. But you also have some variations as you go around the azimuth as well. So it's not quite a simple function of just amplitude. So moving on, this slide has a lot of data, but essentially this is showing for each cell now in the same library, we can see both within cell type across the columns, differences in their spatial sensitivity to the field. And going across layer, we also see differences in spatial sensitivity in the field. But one thing we do see that it agrees with, I think, conventional wisdom is that these pyramidal cells are fairly essentially polarized most strongly for downward and upward fields. Those are fields that are going into and out of the cortical surface. So that was hopefully helpful to orient us as we go into this final stage, which is to then look at how do these neurons polarize in the actual brain in response to, for example, conventional tDCS or a high definition tDCS array. In this case, just looking at a 4x1 ring and positioned over M1. So here I've just simulated these in SimNibs again with currents that produce about matching peak electric fields, but which produce a higher electric field for the HD array in the M1. And so now I'm gonna kind of basically reproduce the plots like before with the thresholds, but looking at polarization. And what we can see is, again, layer-specific effects, but also both, in this case for anodal tDCS, both depolarization and hyperpolarization right underneath the electrodes, you can see with the red and blue regions. And these are pretty easily explained if we just think back to those maps, which is where the field is entering into the cortex, we're seeing depolarization, and where it leaves, for example, the other side of this striral crown, you start to see hyperpolarization for the layer, for the pyramidal cells especially. And what we can also do is quantify not only polarization in the full region, but polarization within subregions. For example, if we identify this kind of putative hand mouse representation we were looking at earlier, we see that the HD tDCS, while it clearly has a much more focal electric field distribution, that's kind of trivial just looking at the electric field, and a higher peak, it does actually produce a wider range of somatic polarization, even in the more selected subregion of the M1, or this hand knob kind of region of interest. So when we talk about whether or not anodal or cathodal tDCS are producing largely depolarization, hyperpolarization, we can already tell from these models that it's much more complex. There's going to be some kind of balance of depolarization, hyperpolarization being produced, especially if we're just looking at the cell body. But what we also can do with these models is look beyond the cell body at the axons and dendrites. So that was kind of a taste of what we can do with these models, looking at tES and the complexity that I think that they bring out in terms of the specifically polarization that's produced. So with that, I think I'm running out of time and I can just conclude. So I hope that I've been able to explain a little bit about how this framework works for simulating the direct effects of tES and TMS in this case. And shown that these kind of models allow you to capture some phenomena that can't be explained by the electric field alone. The models that we've been using have shown that the axonal morphology especially is critical in determining the effect of the spatial sensitivity of these neurons to the electric field. And specifically for TMS, for example, we found that the axon is activated at its terminal, and because of its particular sensitivity to the electric field magnitude, this activation occurs in the superficial gyral crown lip or crown and lip region, which is driven by electric field magnitude. But the kind of subtle sensitivity to the direction produced these direction-specific effects that produce that anterior shift that we talked about. Whereas with tDCS, we see that within a cell, you have this biphasic polarization distribution. So you have both depolarization and hyperpolarization. And that also across the cortical region that's targeted, you have both depolarization and hyperpolarization being produced. And the magnitude of that polarization and the exact sensitivity to orientation depends on the layer, the cell type, and potentially has some intra-cell type variability. So what we can do with these models next is potentially investigate how that polarization is affecting postsynaptic integration, for example, via somatic or dendritic polarization, or presynaptic transmission via that axonal polarization, by introducing ongoing activity into these neurons. So these neurons have all, the simulation results I've shown you today have been all in quiescent neurons and looked at steady state polarization. But what we can do next is then say, can we inject activity into these cells, either synaptically or through current injection, and see how tDCS affects them, their behavior in a region-specific manner and a cell type-specific manner. I've shown mostly all results from a single head model, but what we can do with these as well is study intersubject variability with just embedding these neuron models in more subjects, which is fairly easy with this framework. But this kind of raises some questions about how we can make the neuron models and the physiological response subject specific. Right now, the models are all drawn from low-level data from animal experiments. So there might be questions about how we can adapt them to specific subjects. And finally, these models can serve as a kind of input to more detailed, larger scale, but more reduced network models of the circuit response. So we've only been modeling the single cell response. We know, especially with tDCS, that the effects are going to happen at a more network level through modulation of ongoing activity. So what we hope is that these models can provide more accurate estimates of the actual coupling of the electric field to the network to allow us to then predict the physiological response. So with that, I will end and thank my advisors, Dr. Peter Chov and Dr. Grill. The model code that I presented today is all available on ModelDB and on GitHub. And yeah, I want to thank the organizers for allowing me to speak. I'm really privileged to get to join you all to present some of my work. Thanks.
Thank you very much, Aman, for a great talk. Very interesting, very exciting stuff. I want to ask the audience or the panelists to ask questions. I have a couple, but I would like to open the floor. Okay, let me ask you one question then. I mean, I was fascinated by the plots that you showed, this polar distribution for the somatic polarization. And I was wondering if you have started looking at dendritic or axonal termination plots of the same, with the same structure. Do we still see a validation of the wisdom of the conventional wisdom of the relevance of the direction of the electric field, or is that changing like it did with TMS?
Yeah, so actually I do have those.
Awesome.
I wasn't sure if I would have time. Yeah, I think you don't see. Not sure if you were saying there's some. Yeah, I don't think anything is negated necessarily, but I think you do see differences in, for example, which cell or which layers experience the most polarization. So I don't know if I can flip between them, but the somatic polarization layer 2-3 was experiencing much less. Oh, here we go. Sorry. So this is the somatic polarization here versus the axonal polarization here.
Okay.
So the layer 2-3 pyramidal cells you see experience much higher polarization. But here I'm only showing the peak polarization. So there's potentially some variability within any region here across the 5 cell types and within the actual axonal arbor. So we're just grabbing the peak value of all the axon terminals being polarized by the field. Yeah, so I think there's still some analysis we have to do to really see, piece apart what's going on. But essentially, yeah, the data is there to be looked at.
So based on this work, do you think that when we try to develop hybrid brain models, we're going to have to go into a layer type of modeling and consider these directionality effects with axons and dendrites in order to reproduce really what's going on? Or can we, I mean, what I'm asking is how much can we really simplify?
Yeah, no, that's a really good question. I think our goal is to basically extract from these models the ways to simplify them so that you don't have to run the entire detailed model. So essentially what stimulation parameter sets or basically how can you go from a simulation parameter set to the important metrics that you need, or the interesting metrics. So yeah, I don't think you necessarily need the full detailed model in all cases, but I think it helps us kind of constrain or guide how we simplify, I think, the network models and what we input to those network models. For example, the magnitude of the polarization we can estimate more accurately, and the magnitude of polarization in the axon and the dendrite separately.
Yeah. Absolutely, yeah. And we still don't know which one of those is the most relevant physiologically.
Yeah, I mean, so I've shown data from the sunlight, but yeah, I think my inclination, and I think what I've seen from others is the synaptic effects seem to be most likely the mechanism of action. So yeah, I think the axon dendrite really are the more probably useful place to look.
Okay, any other questions from panelists or audience? We're right on the next transition point. So if there are no further questions,
maybe one
please.
question. Yes, do you plan in fact to simulate unit activities from, microcircuits, and then, for instance, study the effect of the polarization of axon terminals and the effect on the probability of release of neurotransmitters onto the timing of action potentials, and then the spike timing dependent plasticity. Would you be able to do that? Because, and in that case, how would you handle, in fact, the connectivity among these neurons, which is not completely known for the neocortex?
I think that's a big question. I think it's all theoretically possible, or at least it's the kind of framework can handle that type of model. But I would say the difficulty, yeah, exactly, is the connectivity is not clear. The models we're drawing are from a network model. So this Blue Brain project, they built this essentially a cortical column model of a rat's somatosensory cortex. So we can take data from that connectivity, but the detail of these models, the number of compartments simulated, number of channels, makes it difficult to scale up and make full-scale network models with these neuron models without a supercomputer, basically. So I think that's why it's another approach that maybe is more tractable is to feed the kind of low-level metrics into more reduced network models, more high-level model representations of brain function. But I do think that first step, though, you mentioned about studying just the effect on synaptic transmission is definitely doable in these models and something that we're interested in doing. So that would just involve basically adding presynaptic terminal specific channels, like, for example, the calcium channels that we know exist there. And from there, we can actually, I think, estimate the effects on the, for example, effects of polarization on action potential shape, which affects calcium influx, and which would then affect synaptic transmission. So those kind of mechanisms are very, very easy to, I think, capture in this type of model.
Okay, thank you.
Okay, thank you very much. So thanks again, Aman, for this very nice talk. Let's move on up the scale with Stephanie. Stephanie Jones will talk about the Human Neocortical Neurosolver, a new software tool to study the circuit origin of EEG and impact of non-invasive brain stimulation.
I'm still sharing.
Okay, so can you see my screen okay and hear me? Okay, great.
Yes.
So I'd like to start by thanking Julio for organizing this session. I'm very glad to have the opportunity to talk about our new software, Human Neocortical Neurosolver, that's designed to study the circuit origin of EEG signals, and it can be applied to study the impact of non-invasive brain stimulation on neural dynamics and brain function. So I'd like to begin by describing the challenge in human neuroscience that we aim to address by developing this new tool. So we all know that EEG is a very powerful technology because it gives us a direct correlate of human information processing with millisecond resolution. It can also be used as a way to read out the impact of non-invasive brain stimulation on neural circuit dynamics. But the downside is that what we're recording is a so-called macro scale activity that's generated by large ensembles of neurons. And it's still difficult to infer what's going on at the underlying cellular and circuit level. And this circuit level understanding is really critical if we want to be able to use EEG to help us understand what non-invasive brain stimulation is doing, and in fact, to help us ultimately design better simulation paradigms. And so invasive recording techniques are really ideal for dissecting circuit level activity. And there's lots of very exciting advances in this domain right now, but there's still challenges in applying these techniques in humans. And so how do we translate from this signal that we measure outside of the head to what's going on inside of the head? Well, this is the ideal problem for computational neuromodeling, where we can simulate the electrical activity of the neurons. And with models, we can have specificity both at this microcircuit level and at this macro scale recording level. And so this has been the primary focus of my research program. And we've recently turned this modeling framework into a software tool that's being designed in this user-friendly manner for clinicians and researchers to be able to develop and test hypotheses on the circuit origin of their human EEG or MEG data. We call it Human Neocortical Neurosolver, or HNN for short. So in my talk today, I'm going to start by giving you some of the background information on the development of this new neuromodeling tool. And I'm going to go through an example of how it can be applied to study one of the most commonly measured EEG signals, a sensory evoked related potential, or ERP. And I'm going to use an example from my lab where we've been studying tactile perception and investigating the origin of a tactile evoked response. I'll then give an example of how HNN can be applied to study the impact of non-invasive brain stimulation on neural circuits and behavior. And again, I'm going to focus on a study from my lab where we attempted to modulate tactile perception with transcranial alternating current stimulation. So how do we begin to develop a model that can connect these extracranial signals to something that's going on at the microcircuit level inside of the head? Well, there are two things that we need to consider along the way. The first thing is that these EEG signals are created by huge electrical currents in the brain. And the first thing that we need to do is we need to apply inverse solution methods to estimate the location, direction, and time course of these underlying electrical currents that we call primary currents that contribute to the extracranial recorded signal. From there, we need to think about where are these primary currents generated, or how are these generated in the underlying neural circuits. And it's believed that these primary currents are generated by the postsynaptic intracellular current flow in these long and spatially aligned cortical pyramidal neuron dendrites. These dendrites create this big electrical current that produces an electric and a magnetic field that we record outside of the head with EEG and MEG. And again, it's the spatial alignment of these neurons that allows the summed activity across large populations of pyramidal neurons to produce this large signal that we can record outside of the head. So given that this is where the signal comes from, when we want to create a model to study the cellular and circuit origin of this signal, there are some key features of cortical circuitry that we need to account for. The first is that the cortex is a layered structure. It contains various types of excitatory and inhibitory neurons across the cortical layers, including pyramidal neurons that have these extended apical dendrites in both the supragranular and infragranular layers. These networks don't sit in isolation in the awake behaving brain, but they're constantly receiving input from other parts of the brain. There's two primary pathways of input to the local cortical circuit that we need to consider. The first is a pathway that relays so-called feed-forward information. This is the information that's relayed from, say, a sensory input through the thalamus and into the cortical structure through the granular layers, where it then propagates to the other layers. The other pathway of input is what we call feedback input that comes from higher order cortical areas or from these non-specific non-lemniscal thalamic nuclei that project right up to the supragranular layers and contact the distal dendrites of the pyramidal neuron there. Now, again, we can calculate this big electrical primary current signal from the intracellular current flow across all of the pyramidal neurons across the layers. Now, with this construction, the units of measure that would come out of a model like this are current times distance. You have a current over a distance. And that's the same unit of measure that you get from your source localized MEG or EEG data. And so you have one-to-one comparison between the output of the model and your recorded data. Now, we know something about the size of the networks that contribute to some of the most commonly measured EEG signals. Some of the most commonly measured signals are ERPs, or low frequency oscillations. ERPs are typically on the order of 10 to 100 nanoampere meters, and they're thought to be generated by the synchronous activity of on the order of tens of thousands of synchronously firing pyramidal neurons. Low frequency oscillations tend to be even larger. They're on the order of 100 to 1,000 nanoampere meters, and they're believed to be generated by the subthreshold activity of on the order of 1 million pyramidal neurons. And so what I'm showing you here is clearly a reduced representation of the details of the cortical circuitry. And we just saw some nice detailed models in Aman's talk. But when you record outside of the head, you don't have access to all of the finer details of the neural circuit. And there's something meaningful you can say about the origin of the signal with these canonical features. And so this is the level of detail that we built into our model. And I want to just spend one slide giving you a little more detail of what we've simulated in HNN. And so we have multi-compartment pyramidal neurons and single compartment inhibitory neurons. They're synaptically coupled with GABAergic and glutamatergic synapses. And we simulate the electrical activity in each compartment with Hodgkin-Huxley dynamics. We also have cable theory to get this intra-longitudinal current flow within the pyramidal neuron dendrites. We do have active ionic conductances in the somatic and dendritic compartments. And again, we calculate this primary current signal from the net intracellular current flow. Now, as I mentioned, it's important to consider the inputs to this circuit from other parts of the brain. And in our model, we don't simulate the activity of all these other brain areas. We're not doing a whole brain model. Instead, what we're doing is we're generating trains of action potentials in predefined patterns that activate synapses in the local network and these feed forward and feedback projection patterns. So for example, I can simulate a train of action potentials that represents a feed forward drive. These action potentials are going to excite excitatory synapses in the local network on these proximal dendrites that will push current flow up the dendrites and can create this upward deflecting signal. If I have a feedback input where I simulate a train of action potentials that activate excitatory synapses on the distal dendrites, this pushes current flow down the dendrites and can create a negative deflecting signal. And when we have a combination of these inputs in this active cortical circuit, it drives current flow up and down the dendrites and creates the signal that we record. Now, this is a reduced representation of the full network. The full network in HNN contains a scalable number of pyramidal neurons and interneurons in the supragranular and infragranular layers. It represents a patch of cortex in a localized brain area. And so we're looking at a single brain area. And we've designed the software to be able to study the origin of these commonly measured signals, including ERPs in low frequency rhythms. So this is a really complicated, large scale network model. The code's available, it's open source, you can find it on GitHub. But unless you know how to build neural models and adapt neural models, it can be really hard to understand this underlying code. And so one of the things we're working really hard to do is to make this a user-friendly model. And so to do that, we've embedded HNN in a graphical user interface. And so you don't need to do any coding in order to interact with this model. We've created a website that has installation instructions. We provide tutorials on how to simulate some of these commonly measured signals, and we give example data sets and parameter sets from our own studies. And so when you start the software, this is what you see. This is the main GUI window, and you see a representation of the local network. And importantly, these layer-specific proximal feed-forward and distal feedback pathways of input. And so next, I want to give you an example of how you can use this software to study a commonly measured signal and event-related potential. And for this, again, I'm going to go back to studies in my own lab, where we perform tactile detection experiments. We give brief taps to the middle finger of our subjects, and we ask them, did you feel it or not? And we maintain the strength of this tap so that they're feeling it approximately half of the time. We record the brain activity with either MEG or EEG, and then we use inverse solution techniques to isolate the contribution of the signal from the hand area of S1. And so what the signal looks like in the hand area of S1 when we tap the finger is as follows. I've loaded this data into my HNN GUI. And this ERP was generated by a fairly strong tap to the finger that was felt 100% of the time. And so you can see that it creates these clear and reliable evoked response peaks. And so the first study we did with this model back in 2007 was to see, could we use the model to understand how this ERP is being generated by this underlying cortical circuit? And so to do that, we went back to the literature and said, well, what do we know about how ERPs are generated? And it turns out, particularly for somatosensory evoked potentials, there's a huge history of literature that's looked at the origin. And what that literature suggests is that these evoked responses are generated by a sequence of activation to this local cortical network that consists of a feed forward, followed by a feedback, followed by a reemergent feed forward input. And so we built this pattern of input into our local network. And so what I'm showing you on the top here is the histogram of the input patterns to the local network that we're going to simulate. And so this is a spike histogram, and this is the envelopes of the spikes that represent the initial feed forward input that's going to come up through this proximal distal, this proximal projection pathway. Then we have our feedback distal input, and then we have our later feed forward input. And so when I hit run simulation, this model is going to respond to these inputs and produce some kind of a macro scale signal. And this is the macro scale signal that comes out from that pattern of feed forward and feedback input. And you can see that it's very nicely able to reproduce this evoked response pattern. Now, importantly, this pattern of input was motivated by the literature. But we spent a lot of time tuning the network. We tuned the timing and the strength of these inputs so that we could get an accurate representation of this ERP signal. And so now we're at the point where we can ask the question, well, when this signal changes across different experimental conditions, where does that change come from in the neural circuit? And so in our initial study, we looked at differences in sensory evoked responses from taps to the finger that were detected and non-detected. But what I want to concentrate on this talk is that we've also looked at differences in the evoked response that occurred from non-invasive brain stimulation and how we can use our model to understand the underlying circuit differences that create that different evoked response. And so I'm going to move to that next. But first I want to switch to describe the non-invasive brain stimulation study that we applied in the lab, where we tried to use transcranial alternating current stimulation to modulate tactile perception. And so I want to start with, again, some background motivation for the development of this paradigm. In addition to looking at tactile evoked responses, we've been studying spontaneous rhythms and how they impact the ability of a subject to feel the tap to the finger. So here I'm showing you again the activity from the hand area of S1. This is the spontaneous activity before we tap the finger. And you can see that there's this large amplitude, low frequency oscillation. When you put a frequency filter on it, you have activity in the alpha and in the beta bands. But at any moment in time, the power is changing. And so we ask the question, does the power in this pre-stimulus period predict whether the subject will feel that tap to the finger or not? And it turns out that these pre-stimulus oscillations are a very strong predictor of perception, such that the more power you have in the pre-stimulus period, the less likely you are to feel that tap to the finger. And so in this way, these spontaneous rhythms in S1 are a signature of inhibited perception. And we've also seen that you can change the expression of these rhythms with attention, such that when you pay attention, the power decreases. And so we looked at this separately in the alpha and beta bands. And here what I'm showing you is we're averaging this pre-stimulus power across time and across the frequency band, and we're sorting from low to high power. And we're looking at the detection rate, which is a percent change in hit rate from the mean. So the higher it is, the more likely you are to feel the tap to the finger. The lower it is, the less likely you are to feel the tap to the finger. And you can see very clearly that in both the alpha and the beta band, the more power you have in the pre-stimulus period, the less likely you are to feel the tap to the finger. So with this result in hand, we came to the point where we wanted to ask, well, can we modulate these rhythms with non-invasive brain stimulation, and in doing so, causally impact tactile perception? And if so, can we use our HNN modeling framework to interpret the circuit level effects of this non-invasive brain stimulation? And we started by looking at this alpha frequency oscillation. There's again a large history of work that suggests that you can maybe enhance alpha oscillations with brain stimulation, and particularly with alternating current stimulation. And so that's where we started. We developed a study where we were doing this tactile detection task before, during, and after 10 minutes of individualized alpha frequency stimulation over the contralateral primary somatosensory cortex. We had two groups, one that got the alpha stimulation, which was very weak at 1 milliamp, and was on for 6 seconds, then off for 6 seconds. And then we had a sham group that had just this one cycle of a 1 hertz rhythm that ramped up and then turned off. And what we wanted to compare was both the EEG activity in the alpha band and in the ERP before the TAC stimulation and after the TAC stimulation. And we also wanted to see, did TACs impact perception? And so again, our hypothesis was, well, maybe we can enhance this alpha oscillation, and in doing so, inhibit tactile perception. And what we found was as follows. We didn't find that this stimulation paradigm impacted the alpha oscillations in any way. And so here I'm just showing you the pre and post conditions in both the pre and post alpha power in the sham and alpha stim conditions. And you can see there's no difference in alpha power. We also didn't see a sustained difference in performance. And so during the alpha stimulation, we did see effect of alpha stimulation compared to sham on performance. And so again, this is the percent change in that hit rate from the pre-tax baseline. And you can see that with the alpha stimulation, performance went down. But this didn't persist into this post-stimulation period. Now, there are lots of reasons why we may not have seen changes in alpha and perception in our study. There were a lot of methodological issues with this design. It was a very weak stimulus, and we talk a lot about that in our publication on this work. But what we did see was a subtle but significant effect on the tactile evoked response, such that in the post-alpha stimulation condition, we got a larger amplitude peak near the 70 milliseconds response. And this was significantly different when compared to the sham condition. And so what we said was, maybe we can use our modeling software to understand how it is that this evoked response emerges. And so here we turn to our HNN software. And again, we simulated the ERP like I showed you before, and we've titrated the strength of the input so we get an accurate representation of this pre-tax condition. Now, there's a lot of things that might be happening in the local network to create this difference that we saw after the stimulation period. But there was a specific hypothesis that was perhaps most likely based on the literature, and that's that maybe this alpha frequency stimulation is changing the local synaptic plasticity in the network. And so we have these local excitatory and inhibitory connections, and we can go into the model and change those connections and see, does that change account for the difference that we see in our data? And so we've created a way to do this easily in the model, where you go into the set parameter buttons, and we have a button here that's called synaptic gains. And with this, you can change all of the conductances of the excitatory inhibitory synapses with just changing this one variable, which represents a multiplication factor to the strength of that input. And so you can do this for the excitatory inputs, you can do it for the inhibitory inputs. And so the first thing we did was say, let's increase the strength of these synapses twofold, right? And so we're going to increase the maximal conductance by a factor of 2. We rerun our simulation and we see what does that do to the output of this network. And increasing the synaptic gain of all the neurons did reproduce this difference near the 75 millisecond peak, but it also reproduces this later difference that we didn't see in our experimental condition. And so then we went in and said, well, maybe the stimulation, the alpha stimulation, was differentially impacting the excitatory or the inhibitory cells. And so we said, what if we just change the synaptic connection among the excitatory, from the excitatory cells? And what we found was actually that goes in the opposite direction. It makes a larger amplitude signal overall. And then we tested, well, what if we do only the inhibitory neurons? And with only changing the inhibitory synaptic plasticity, we were able to reproduce this downward deflection that persisted throughout the evoked response period, similar to what we see in our experimental design. And in fact, this fits with some of the literature that suggests when you give very low intensity alternating current stimulation, here it was 1 milliamp, this may preferentially potentiate inhibitory synapse. And so it is consistent with the literature. And so the model was nicely able to reproduce the data and provide some, before I go on to my next slide, there's one thing that I wanted to show. One of the powerful things about the model is that you can go in, this is the macro scale signal, but we can go in and look at all the micro scale activity. And so you can easily do that. You go to view and we can look at various things in the underlying simulation. Here I'm going to look at the spiking activity in the individual neurons. And so this is a raster plot of the underlying spike trains during this ERP. And we see the spike trains of the inhibitory neurons, the excitatory neurons in each of the different layers. And what we see is that during this prominent 70 millisecond peak, when we have this increased inhibition, we get less spiking in the pyramidal neuron populations. And this corresponds to a smaller amplitude signal in our macro scale simulation. And so while this alternating current stimulation isn't driving spikes, it's changing the synaptic efficacy in the local network such that when we give that tap to the finger, it drives spikes differentially to create that difference in the evoked response that we recorded. Okay, so we were able to reproduce our data. We have some really targeted predictions about what's going on in the underlying circuit that then we can further test with invasive studies. But one of the goals of our study was to modulate perception. And with this experimental paradigm, we didn't modulate perception. So from here, we went back to look more closely at our data. And it turns out in this data that the beta activity is in fact more prominent and is a stronger predictor of perception than the alpha activity. So we set out to see if we could instead modulate the beta rhythm with brain stimulation. But before doing that, we took a step back to understand what our data looks like when it's not averaged. What I'm showing you here is activity averaged across subjects, across trials. Here it's averaged across time. But we're giving our tactile stimulation, the brain is not averaging. And so we wanted to see what is the signature of this activity in the unaveraged data. And it turns out that these beta oscillations are very transient in time. And so here I'm showing you 10 examples of unaveraged data. This is the raw waveform, and here's the corresponding frequency spectrum. These beta oscillations emerge as these transient events. And because in the spectral space, the signal is always positive, when you average this signal, these events accumulate without cancellation. And you get this representation that there's this continuous oscillation in the brain, when in fact, it's a very transient signature of activity. Now, this transient nature of beta oscillations is a very robust phenomena. We've observed it in different recording modalities, different experiments. We observed it in local field potential recordings in mice. It's been observed in motor cortex, in striatum, and so it seems to be a very robust phenomenon. Now that we know that these rhythms aren't continuous, we have to revisit the question of what creates high power in the signal. Now, typically when we think of high power, we think of an oscillation that's higher in amplitude and a larger network is contributing to the response. But now that we know these oscillations are transient, there's several features that can contribute to differences in average power. It could be, so this is just a schematic representation on a trial where we could have low mean power if we average across time in the frequency band, or high mean power. It could be that on some trials we have one event, and on other trials we have two events of the same amplitude. And we average this, we get low power, and that we get high power. It could be that the events actually are higher in amplitude, and so that when you average, you get higher mean power. It could be that they last longer, or it could be that the frequency changes. Any one of these could underlie the difference in power that we're seeing in our data and could be functionally relevant. So it turns out that it's actually the number of these events that contributes to the differences in power that we're seeing and contributes to inhibited perception. And so that on trials where the subject doesn't feel the tap to their finger, there's more of these events. And the timing of the events also matters, such that the closer they occur to when that tap occurs, the more likely they're inhibited detection. And so now we're at a point with this information where we have to rethink about how we design our stimulation paradigms. And it's led us to this question of how might we apply non-invasive brain stimulation to more accurately account for these transient brain rhythms? And in doing so, we might more accurately reflect what's going on in the brain and be able to more effectively inhibit perception. And so with this information, the next steps that we're working towards is to think about how we can develop non-invasive brain stimulation paradigms aimed at mimicking this endogenous expression of transient rhythms in the brain. Currently, we're planning to do this not with alternating current stimulation, but with TMS, where we can have more control over the pattern and the timing of the inputs. We also now want to think about integrating our HNN neuromodeling framework with the field modeling frameworks that we've been hearing about in this session to have an improved understanding of how the connection between the two works. So with that, I'm going to end, and I have conclusions here that the human neocortical solver is this neocortical column model under thalamic cortical and cortical drive. We're distributing the software through a new website, which has all sorts of information. And I hope I've shown you that it can be applied to understand the impact of non-invasive brain stimulation on circuits through their corresponding changes in EEG, and can aid in the development of new non-invasive brain stimulation paradigms. And so I apologize, I think I've gone over, but I of course want to thank my collaborators. HNN is being developed by a large collaborative team with researchers at Brown, Mass General, Yale University. And this is just a picture of our development team. I also want to give a special thanks to Danielle Sliva, who's a graduate student in the group. She's the one that's been leading our non-invasive brain stimulation studies and perform those modeling experiments for our alpha stimulation work. And so thank you for your time. And again, my apology for going a few minutes over.
Thank you very much, Stephanie, for a wonderful presentation. Very exciting stuff. Any questions from the panelists while you guys think about it, or girls? I have one question. I mean, I think it's very interesting that you're bringing together brain stimulation and these detailed models. And you showed in your model of ERPs that by tweaking inhibitory connectivity, the gain of the synaptic inputs, you could reproduce better data. Have you thought about how this change in connectivity, this plastic change, so to speak, could come from the interaction of tCS with the model? Have you tried to model that yet? I mean, in light especially of the nice work that also Aman showed.
Yeah, we haven't yet coupled our neural model with a field model, but that is something that we want to do. Instead, what we do is we make predictions about, well, what happened to create the difference that we saw. And so we just go in manually and say, well, we are going to assume there's a change in plasticity, and we can simulate that by changing the connectivity strength within our network. But we're just hand tuning that knob, and we're not yet coupling this with the actual brain stimulation field modeling. But that's a direction that we want to head in. It's obviously important.
Right. I have a question here in the Q&A for you from Philip Sabes. Great talk, thank you. How unique are the solutions to the ERP responses with respect to the parameters adjustment, I suppose? And how sensitive to the solution would be the model conclusion about what the effect of tACS is?
Okay, so for the first question, can you repeat that first question again, Julio? I just want to make sure I answer.
The first question is how unique is the solution, the parameter feed that you show that gives you the best feed to the data? Okay. How unique is that with respect to the parameters that you can adjust?
Yeah, that's a really good question. Again, this is a large scale model. There's lots of parameters here, and there's lots of differential equations. And the process for us in simulating the evoked response was sequential. And so we didn't start by saying, well, can I change every parameter of the model and how will that reproduce the ERP? What we did was we first created the individual.
Sorry, Stephanie, we just lost you, I think. Let's see if you come back. We just lost Stephanie, I think. Can the rest of you hear me okay? Yes. Okay. I think we just lost the connection. So in the interest of time, I think we're gonna move on because we're a bit late. Perhaps if she comes back online, we can go back to this question in a moment. So the last question, the last presentation, sorry, is from Fabrice Wendling from INSERM, a great friend and collaborator of mine for many years now. He's going to be talking about the mechanisms of action of tCS in epilepsy and the need for hybrid bio-inspired brain models. So Fabrice, you want to take the floor, please? Okay, Fabrice, go ahead, please. You're muted, but you can start a presentation.
Can you see my screen now? No. Can you see it?
Yes.
Okay.
We can see and hear you.
Okay. Okay, so today we are going to talk about tCS and epilepsy. So as you will see, there are a number of studies that have reported therapeutic effects of tCS in epileptic patients. But as you will see, I mean, the situation is not so easy. It's a complex problem to optimize, in fact, the stimulation in the context of epilepsy. And what I will show is that we need, in fact, models that combine both the biophysics and the neurophysiology in order to move forward. So just to remind you that tCS is a modulatory technique, okay? It's only a fraction of current that reaches the cerebral cortex. And when you think about TMS at the level of neurons, it's only an electric field of the order of 50 to 100 volts per meter that can trigger an action potential, okay? The electric fields that are induced by tCS are much lower, something like 50 to 100 times weaker. So we are dealing with weak electric fields. So the order of magnitude of the electric field at the level of the cortex is only to 1 or 2 volts per meter. So tCS is a neuromodulator, let's say, involves neuromodulation and not neurostimulation. So, and this makes the problem even more complicated since it's a subthreshold perturbation of the brain. So as I said in the introduction, there are a number of papers that have been recently published, in fact, on the treatment of epilepsy using transcranial direct current stimulation, tCS. So for instance, in the Lennox-Gastaut, in pharmacoresistant epilepsy. So there are a number of papers. There are also review papers that say that neurostimulation is a promising therapy for epilepsy, and especially non-invasive brain stimulation, okay, for the treatment of epilepsy. So there are, if you look at the literature, there are now quite a number of not only original papers, but also reviews. There are some papers which provide positive results, I would say. For instance, this one, transcranial direct current stimulation for the treatment of refractory childhood focal epilepsy. So they used a specific protocol. I will just show you, the main results. There were two arms, in fact, in this study. 27 kids were stimulated using actual stimulation, tCS, and the other arm was 9 children receiving sham stimulation. And they just, in fact, counted the number of epileptiform discharges at the level of the EEG in the two groups. And as you can say, okay, in the group of children receiving the actual brain stimulation, there is a significant reduction of epileptiform discharges. And very importantly, this reduction tends to last in time and is still significant 1 day or 2 days after the 20-minute stimulation session. So there are definitively papers, even review articles, showing that indeed tCS may decrease epileptic seizures, okay, and reduce epileptiform activity. Unfortunately, there are a number of papers showing that these effects are not significant. Typically, I will not go in the details of that study. They are using multiple sites or high definition tCS in focal epilepsies, drug resistant, and just look at the conclusion. The effects, okay, and typically the changes in epileptiform discharges or the mean seizure frequency is not statistically significant between two groups, okay, sham versus actual stimulation. And in this recent paper, which is a review article, okay, in the conclusion, one can read that there is no recommendation that can be made about a potential efficacy of tCS in the treatment of any type of epilepsy. So the situation is complex, and I think that it's probably due to many factors or reasons that I've tried to summarize on this slide. When we look at the literature, there is a heterogeneity of patients and of forms of epilepsy. We know that epilepsy is not a single disease, it's a syndrome, okay? And it's a syndrome that may take different forms with different etiologies, okay? Also, the protocols are not, let's say, homogeneous. There is a multiplicity of protocols. The duration is important, the intensity of stimulation, the montage that is used for stimulating the brain, and also in the case where sessions are repeated in time, the number of sessions might be an important factor, also explaining the difference between studies. Another, let's say, parameter is the definition of the readout. Okay, some studies quantify the seizure frequency, the other it's more the markers that we get before and after the stimulation, and which may be quantified using different features. What is to me important is probably also the absence of account for the patient specificity. And this deals with precision medicine. Probably if we want to make more progress on the use of tDCS in the field of epilepsy, we must in fact account for the specificity of each patient, because epilepsy is a neurological disorder which is relatively specific to the patient and which strongly differs, in fact, from one patient to another one. There is also a plethora of mechanisms which possibly lead to epileptic or epileptiform activity and to seizures, of course. And actually, at present, in fact, there is a lack of knowledge, in fact, of these mechanisms and also about the mechanisms of actions of tDCS onto the epileptogenic network and systems. So what we think is that hybrid models, models that will combine, in fact, the biophysics, distribution of electric fields in the brain, with the neurophysiology, the response, in fact, of neurons and networks to the electric fields, may help us, in fact, to define better protocols for, let's say, optimizing the therapeutic effects. So this is what we plan to do in this project that will start very soon. It's an ERC project funded by Europe. And our intent, in fact, is to try to control epileptic brain networks with weak electric fields that are computationally optimized using computational models. So the objective is relatively simple, okay? What we want to achieve is to, let's say, define some non-invasive brain stimulation protocols, in fact, to, let's say, suppress epileptic activity. And the approach that we will follow is multiple scale. What we want to do is to very accurately model the electric fields that are induced by the scalp electrodes in a patient-specific context, of course, and to combine, in fact, these biophysical models with neurophysiological models that will account for the effect of electric fields at the level of the cells, the neurons, at the level of microcircuits, typically detailed networks of neurons, and also at the level of populations of neurons, typically this mesoscopic level, and then move toward the effects of electric fields at the level of large-scale networks. And this is particularly important in the context of epilepsy, especially for those drug-resistant epilepsies that we target, which very often involve, in fact, large-scale brain network, epileptogenic networks. So for the distribution of the electric field, we will use, in fact, multi-site tDCS, high-definition tDCS. And this is what Giulio, in fact, and his team has been developing for a long time now. The idea, probably you are familiar with this, but the idea is very simple. We want to target one particular region of the brain or a set of regions. And the idea is to optimize, in fact, not only the position of anodes and cathodes, but also the current intensity values, such that the topology of the resulting electric fields match with the targeted regions. We also want to develop neurophysiological models to account for the mechanisms of actions of tCS. And I mean, it's been known for a long time now that there are two types of effects. There are acute effects, okay, immediate effects, I would say, and also lasting effects, after effects, okay. Acute effects are very likely dependent on membrane polarization, as Aman has very nicely shown. And after effects, I mean, have been reported since a long time, and they are probably related to neuroplasticity. I took this paper from Bidman in the mid-60s, and at that time, I mean, the authors already reported that a low magnitude alteration of the physical environment of certain neurons lasting for a few minutes may cause a prolonged change in their rate of activity. So basically, if we try to look a bit more in details in these two types of effects, so immediate effects, in fact, include membrane polarization, especially at the level of axon terminals. And there are a few papers showing that probably there is a significant effect at that particular level of microcircuits. And if we modify, in fact, the membrane potential at the level of axon terminals, it's very likely that we modify, in fact, the probability of release of the neurotransmitters, and thus the timing of action potentials. And if we modify the membrane potential at the level of synapses, very likely we also modify the amplitude of the postsynaptic potentials. There are less studies, in fact, I mean, focused on the lasting effects. What is very well admitted now is that it relates to synaptic plasticity. I mean, one immediate effect of changing the timing of action potentials by modulating, in fact, the resting membrane potential of neurons is that we modify the spike timing dependent, we induce a spike timing dependent plasticity. And if we modify, in fact, the amplitude of postsynaptic potential, it's very likely that we induce long-term potentiation or long-term depression. So in the second part of this talk, I would like to show you some work that we've done using the in silico models, the neuronal population models, in fact, to get some insights onto epileptiform activities recorded in mice, and to get some insights on a protocol of stimulation that could be used, in fact, to achieve some therapeutic effects. So this is this study. What we wanted to achieve is a model-guided control of hippocampal discharges, so discharges in the hippocampus, using a local direct current stimulation. Local direct current stimulation has been extensively studied in vitro. We know that it induces membrane polarization, which are very well described now. Unfortunately, there were very few studies in vivo. So in this context, our objective was to study or to explore in vivo the effects of DC stimulation, but at very low intensity, as you will see, at an intensity that generates an electric field which is compatible with tCS. Just notice that in this case, what we do is invasive stimulation. It's not non-invasive. And to do that, I mean, the approach that we followed is a combined approach in silico in vivo. So what we do is that we use a computational model of the hippocampal activity. This is a model that I've proposed a long time ago. And in parallel, we perform, in fact, experiments. And these were performed in mice. So in this model of epilepsy, animal model of epilepsy, animals experience hippocampal paroxysmal discharges. So these are epileptiform discharges recorded from the hippocampus. So what we do basically is that we simulate HPDs. We reproduce those HPDs in the model, and then we study the impact of neuromodulation at the level of the main pyramidal neurons or the interneurons to derive some results on either the increase or the decrease of HPDs in silico. And then we run the experiments to validate, in fact, the model predictions. So just a few words about the kainate model of epilepsy. What we do is a microinjection of kainic acid in the dentate gyrus. So it's a well-known model for temporal lobe epilepsy. And what is known is that after a period of 2 or 3 weeks, 4 weeks, the animal becomes a chronic, develops a chronic epilepsy, which is characterized by epileptiform discharges in the hippocampus. And epileptogenesis is a long-term process. So basically what we study is those markers of the epilepsy, these called hippocampal paroxysmal discharges. So to do the modeling of these discharges, we use, in that study, we used a neural mass model, which includes pyramidal cells, basket cells, OLM cells. And basically, we have collateral excitation on interneurons, and fast inhibition, slow inhibition. And we extended this model, in fact, to reproduce not only the hippocampal paroxysmal discharges, but also the interval in between. So this is an example. This is a real EEG signal recorded in the mouse, and this is what we simulate with the model. So in fact, the model reproduces not only the shape of the HPDs, but also their duration and also the interval between them. Okay. And this was obtained by changing, in fact, the input to the pyramidal neurons in the model. We also, in fact, implemented the local stimulation effects. Okay. Basically, in fact, we put the electrode such that we achieve either a hyperpolarization or a depolarization in CA1. Okay, the pyramidal cells, but also in the dentate gyrus. We also assumed that the stimulation, the local low-intensity stimulation, not only affects the pyramidal cells, but also the local interneurons. So we have parameters, in fact, to weight the amount of membrane polarization onto the 3 types of cells. So this is what we got, in fact, in terms of electric field distribution. So we put electrodes, in fact, in CA1, CA2. As you can see, in fact, the electrodes are the cells. The main cells are in the opposite direction. So on both types, we can achieve either depolarization or hyperpolarization. And the order of magnitude of the electric field is a bit high, in fact, compared to tCS, but still relatively low with direct stimulation, compared to direct stimulation. So these are, for instance, the model predictions that we got. Typically, so we have these 3 constants, in fact, to weight the impact of the stimulation onto the different types of cells. So the y-axis is the effect on pyramidal cells. The x-axis is the effect on the interneurons. And the blue color, in fact, is the low, corresponds to low duration epileptiform discharges, while the red color in fact, corresponds to very prominent hippocampal discharges. So what we want to achieve is the blue, what we target is the blue effect. So what we got, in fact, is this result. In fact, to go from the red, so long duration HPDs, to the blue, it's better to do cathodal. So meaning that the negative electrode would be in CA1. So this is an example, in fact, of the simulation. So typically, this is the control condition. So the model generates HPD-like discharges. And when we do cathodal stimulation, as you can observe, there are less HPDs in the local field potential, but on average, they are also shorter in time. And this is what is summarized in this box plot. What we got is, in fact, is a lowering of HPDs for cathodal stimulation, hyperpolarizing, while the model predicts, in fact, an increase when we do another. It's the blue arrow here. So we wanted, in fact, to validate this model prediction in mice. And this is what we do. For 2 hours, in fact, we record the baseline. And then for 1 hour, we stimulate 6 times. 6 times 11 times 35 seconds. So this corresponds basically to a cumulated duration of stimulation of 5 minutes for the 60 minutes. Okay. And 6 minutes to the, I'm sorry. So we stimulate every 5 minutes, okay? And the cumulated duration is 6 minutes and 25 seconds. So 10% of the whole duration. So we do cathodal stimulation as predicted by the model, and at a very, very low intensity, 1 microampere. So these are 2 examples of mice, okay? This is before stimulation, and this is after stimulation. And as you can see, even by visual inspection, you can see that there are less, in fact, HPDs right after the stimulation. So this can be quantified, of course. In this mouse, as predicted by the model, we do have a lowering of HPDs for cathodal stimulation, but we did not observe in the experiments the increase of HPDs when we do another stimulation. And this is for the results are relatively consistent from one animal to another. So let's look at the group, in fact, results. No, these are the individual results. So these are results for 5 mice. As you can see, in fact, in some animals, let's say in all animals, during the direct current simulation, there is a lowering of the HPD duration, and this is consistent, okay, in all animals. What we also have is in 3 animals, what we observed is a lasting effect. So meaning that when we switch off the stimulator, in the hour that follows, in fact, the stimulation session, there are still less HPDs, which last also less in time compared to the baseline. But in some animals, amazingly, and we don't have an explanation for that, when we switch off the stimulator, then we go back to baseline relatively rapidly. So these are the group average over the 5 animals. Okay, so what is very consistent is the the drop of the duration of HPDs, okay, during the stimulation. And of course, on average, there is a lasting effect with less HPDs than compared to the baseline. Okay. So just before I finish, I want to show you some examples of local field potentials recorded in mice. Typically, this one, I mean, these animals have a lot of hippocampal discharges, and they are completely asymptomatic. These are not seizures, okay? These are interictal activity, and they last for, let's say, 10, 15 seconds, and they occur several times per minute, okay? During the stimulation, okay, we stimulate every 5 minutes. It's very surprising that the HPDs, they just disappear. So we do, I mean, this low intensity stimulation, okay, which induces a low magnitude electric field, definitively has an impact on this system, okay, involving the dentate and the hippocampus. So what is the take-home message? It's not difficult to convince you that a computational model can lead to efficient neuromodulation protocol. This is what we think. Efficient means reducing epileptiform activity. In mice, what we observe is that at very low intensity, which induces an electric field from 2 to 10 volt per meters, we could achieve such an effect, okay? And our hope, of course, is to elaborate such protocols for patients, okay? So to us, transcranial, non-invasive brain stimulation in general, but maybe transcranial current stimulation in particular, may have a high therapeutic potential, but there is still a long way to go. And we definitively need hybrid models, okay, combining neurophysiology and biophysics, personalized for patients, and also including multiple levels to go beyond and progress. Okay, so this was my last slide. Thank you for your attention.
Thank you very much, Fabrice. Thank you also for being patient and being the last one. I would like to open the floor to questions from panelists or speakers. One question that I can throw in your way is something that we have discussed, and maybe you want to comment a bit further on what is the best path to travel from the microscopic to the mesoscopic scale that we seem to need in order to work with tES fields, which are mesoscopic in nature?
Okay, so it's probably it's a difficult issue. What we think is that we must also develop what we call across scale approaches. And typically what we do in the team, I think I've prepared a slide for doing this. We've just proposed very recently let's say a detailed model, a network model, okay? So we model the activity, so these are biophysical models, okay? And so we model the activity from 10,000 neurons, okay? So not only we include principal cells, pyramidal cells, but also parvalbumin positive, somatostatin positive, vasointestinal positive, also positive. So 3 types of interneurons, okay? And what we do is that we simulate the activity of this network under the control condition and also under tCS. And we have included the many effects of tCS, including plasticity in this model. So what is interesting is that from this model, we can generate local field potentials as well, okay? And this is typically what is being simulated by the neuronal mass model, Okay, so we do have a level, the local field potential level, at which we can compare, in fact, the results we get from very detailed models and more macroscopic models. This is one thing. The second thing is that what I personally think is that a very important feature of neural mass model is the input-output functions. Okay, typically the firing rate as a function of the mean membrane potential. Okay, and what is a bit misleading is that most of the people use the same input-output functions, in fact, for the pyramidal cells and for the interneurons. And this was proposed, in fact, a long time ago by Walter Freeman and Fernando Lopez da Silva and so on. Okay. What we do, in fact, now is that we use detailed models to derive the input-output functions of the population. Okay. And this is what we use, in fact, in the neural mass. Okay. So this is also a way to establish a relationship between what we get from the detailed model simulations and what and a feature that we can implement in the neuronal mass model. Okay, so this is a way. This is an approach across scales, and how we can use, in fact, the knowledge we have in detailed models to put in the more mesoscopic models. This is an example.
Okay. Thank you. And to play a little bit the devil's advocate, what can we not capture with mesoscopic hybrid brain models that is crucial for tES applications such as epilepsy, if anything?
You know, in the neural mass model, we don't have an explicit representation of each neuron. Okay, and of course, we don't have an explicit representation of what happened at the level of a single synapse. And as Aman has shown, and what we also have shown in this paper, is that probably tCS, tDCS, tSES has an impact, I mean, has a prominent impact on the membrane polarization presynaptically. Okay, and this probably impacts the probability of release of neurotransmitters. And typically, it's very practical, in fact, to have an explicit representation of that particular single synapse to account for the tDCS effect. Okay. So this is not, in fact, represented in neural mass models. So we will have to find let's say, a proxy of that particular effect to go to let's say more integrated models, and then to large-scale networks. So I would say, more generally speaking, to answer your question, all these features that are very closely related to the cells, typically, Aman has also mentioned, in fact, the calcium effects, are not explicitly represented in neural masses. And this is where, in fact, the problem becomes difficult, because it's not obvious, in fact, in the neural mass to find so in the neural mass, we deal with macroscopic parameters, and it's not really obvious to establish a relation with much more microscopic parameters. So I'm not saying that it's not possible, in fact, to capture, but we have to find how, in fact. Okay, how do I implement in a neural mass the effect of tDCS at the level of the axon terminals? And this is a question. Okay. Yeah. It's a bit the same for plasticity. Okay. How do I implement spike timing dependent plasticity in a model which does not generate any spike? You know, a neural mass does not generate any spike. So we don't have the spike timing. Yeah. So how can we capture the spike timing dependent plasticity? So, you know, but I mean, by comparing the two scales and establishing relations between the two scales, I'm not pessimistic.
Yeah, we just invent new ways to represent those phenomena in such mesoscale models. I guess maybe it will be possible. Any other comments or questions from the panelists?
Yeah, I have a question. Maybe for everyone, actually. I'm curious what your thoughts are on how to make the models of the neurophysiology more subject specific. So, with the volume conductor models, we have, the source data being specific to the subject, but with the neuron models, we're drawing data from a lot more sources. So what are your ideas, I guess, for doing that? Something that we're thinking about as well.
Probably Giulio can answer this question much. Probably you should answer because modeling. Each patient has a very specific brain. So each human has a very specific brain. So using MRI data and going to patient-specific 3D meshes for the volume conductor and so on is a first way, in fact, to account for the patient specificity. Go ahead, Giulio. I mean, I'm sure that.
No, I mean, what we're thinking about is using EEG as a way to collect information that similar to what Stephanie was discussing before. I mean, using this electrophysiology data to try to fit the models. We can ingest probably other data like TMS EEG. We can think about resistive functional connectivity MRI. Here, Gustavo, I'm sure, has many comments. So we can try to integrate all those things and firm up some of the parameters in the model. Yeah. I don't know if one of you, Stephanie, you want to comment?
One other thing we might think about doing in tuning our models is seeing how the individual brains respond to stimuli, right? And I don't mean brain stimuli, I mean sensory stimuli, right? For example, in our hands, we're looking at somatosensory cortex. How my somatosensory cortex responds to a tap to the finger may be different from yours. And so I can start to tune the parameters of my model to have an accurate representation of your response compared to the other response. And now I've got a different baseline state at which whatever I do next to this network, it's going to speak from a different baseline model that was tuned to your actual response to stimulation. That's just another way to think about how we might begin to tune some of these models where we don't have all the biophysical detail that we want in humans.
Gustavo, you want to comment from the angle of criticality here and entertain those ideas a little bit?
From the angle of criticality, but from the angle of more large-scale level. I mean, the plan. I mean, actually, Fabrizio and Giulio, I mean, in Marseille, Victor Schirza is trying to go exactly in that direction, and I'm also involved in the framework of one of the projects, the European project. I mean, and the idea is really to individualize the model by individualizing, of course, the fMRI, TTI, and intracortical EEG in our cases. And that's, of course, I mean, you don't get the particularities and individuality at the microscopic level, but at least at the large scale level, which could be relevant for epilepsy in the sense that sometimes epilepsy is not only just a local has a local character, but could be caused at the network level. And for those cases, perhaps this individualization at this large scale level could be relevant.
Yes.
That is doable, of course, at that level.
Fabrizio, do you want to comment about the use of SEEG in this case? That's one of the things that you have in mind.
It is obviously also a modality that we can use, in fact, to make models more specific to patients, because I mean, I'm talking about epilepsy. We know that each patient has a very, very specific, let's say, seizure pattern. You see? So if we use, in fact, the depth EEG signals to, let's say, to identify the model parameters, the model will become itself more specific to the patient, okay? If the model parameters are tuned, in fact, to reproduce that particular specific pattern of seizure or of interictal activity, then the model will become, in fact, much more specific to that particular patient. Okay, so the more we will use, in fact, data recorded in each single case, the more, and we use this data, in fact, to identify the model parameters, the more the model itself will be a model of that particular patient. Yeah.
One thing that we perhaps haven't talked about very much is about how to bridge the metabolic gap and make use. I mean, we have, Gustavo has talked about the use of fMRI data, but I think we need some modeling work there too as well to make those two worlds come together closer, no? Electrophysiology and these neural mass models with metabolism, vasculature, and so on.
Yeah.
Not much on that direction, actually.
Yeah.
Every model that we have of brain stimulation is thinking about the electrical or magnetic fields hitting the neurons. But there's all sorts of vessels and glia and all other things that are being modulated by these that we haven't yet even started to consider in building our models of how these things impact the brain. And so I think that is really what we should all be doing, is thinking about these non-neuronal systems and how they're being plastically changed by, 10 minutes of some kind of simulation or more.
There was this paper, in fact, published, I think it was 2 years ago in Frontiers. And what they show, in fact, is that, I mean, you can read the conclusion of that paper. I can send you the reference if you want. What they show is that now it's very difficult, in fact, to exclude a contribution of glial cells, in fact, I mean, from the effects of tCS. What they show, I don't know how they say exactly. I don't remember how they. But
it's a modeling
they show that probably tCS or tCS has an effect on glial cells. Yeah, and then on neuroplasticity, because we know that I mean, microglia and they I mean, probably impacts also the plasticity.
Okay, very good. Any other questions or comments? We're a bit late, but it's too fun.
No, it was interesting. Yeah. It was
okay.
Thank you for organizing that.
Thank you very much for your patience with the medium. I think we're learning how to use it, and more or less it went to work well. So thank you again for joining me and joining our panelists, and have a good evening, morning, whatever it happens to be.
It's going to be an evening for us.
Thank you.
Myself.
It's about lunchtime here, so.
And I can tell you that quarantine will probably end by mid-May in France. So it's still the lockdown. Yeah.
Yeah, we still have to be patient. That's
yeah, clear. Yeah. I don't know how it is in your country, but probably it differs from one state to another.
I think we're behind. I think here we're behind, right, Stephanie? It's a bit.
Yeah. We just got word that
up, so to speak,
canceled through the rest of the year for our kids, so that was an interesting development. Kids will not be going back to school.
Okay.
All right. Take care, everyone.
Take care.
Thank you again.
Thank you.